the human genome. It's a term we hear frequently, often in the context of groundbreaking medical advancements or fascinating insights into our ancestry. But what exactly is the human genome? And why is it so incredibly significant? In the simplest terms, the human genome is the complete set of genetic instructions that define a human being. Think of it as the ultimate blueprint, a comprehensive manual containing all the information necessary to build and operate a human body. It's the biological instruction manual passed down from generation to generation that dictates everything from the color of our eyes and hair to our predisposition to certain diseases. This blueprint is encoded in deoxyribonucleic acid, or DNA. DNA is a molecule shaped like a double helix, often described as a twisted ladder. The rungs of this ladder are formed by pairs of chemical bases: adenine, a cytosine, C, and guanine, G. These bases always pair up in a specific way. A always pairs with T, and C always pairs with G. This pairing is fundamental to how DNA works and how genetic information is passed on. The sequence of these bases, the order in which A, T, C, and G appear, is what contains the actual genetic information. Just as the sequence of letters in a word determines its meaning, the sequence of bases in DNA determines the instructions for building proteins, the workhorses of our cells. Proteins carry out a vast array of functions, from catalyzing chemical reactions to transporting molecules to building cellular structures. The human genome consists of approximately 3 billion of these base pairs. To put that into perspective, if you were to type out all 3 billion base pairs, it would take you over 50 years of non-stop typing. And that's just the raw sequence. The real complexity lies in understanding how that sequence is organized and how it's used. Within this vast sequence of base pairs are genes. Genes are specific segments of DNA that provide the instructions for making a particular protein or performing a particular function. It's estimated that the human genome contains around 20,000 to 25,000 genes. While that may seem like a lot, it's surprisingly fewer than some other organisms, like rice, which has over 40,000 genes. This highlights that complexity isn't always about the sheer number of genes, but also about how those genes are regulated and interact with each other. These genes, along with the rest of the DNA, are organized into structures called chromosomes. Think of chromosomes as the chapters in our instruction manual. Humans have 23 pairs of chromosomes. For a total of 46, we inherit one set of 23 chromosomes from our mother and one set of 23 from our father. One of these pairs, the sex chromosomes, determines our biological sex. Females typically have two X chromosomes, 20, while males typically have one X and one Y. Chromosome X, Y. The discovery and mapping of the human genome represent one of the greatest scientific achievements in history. The Human Genome Project, an international effort launched in 1990, set out to determine the complete sequence of the human genome. The project was completed in 2003, providing scientists with an unprecedented level of detail about our genetic makeup. But the Human Genome Project wasn't just about sequencing the DNA. It was also about understanding the function of genes, identifying genetic variations, and developing technologies for analyzing and manipulating DNA. This has paved the way for a revolution in medicine, biology, and our understanding of what it means to be human. So, why is the human genome so significant? First and foremost, it provides a fundamental understanding of human biology. By studying the genome, we can learn how our bodies develop, function, and respond to the environment. This knowledge is essential for understanding the basis of health and disease. Secondly, the human genome is transforming medicine. By identifying genes associated with diseases, we can develop new diagnostic tools, personalized treatments, and preventive strategies. This is the promise of personalized medicine, where treatments are tailored to an individual's genetic makeup. Thirdly, the human genome provides insights into our ancestry and the evolution. By comparing the genomes of different individuals and populations, we can trace our origins, understand how we've adapted to different environments, and learn about the history of our species. Fourthly, the human genome is driving technological innovation. The technologies developed for sequencing and analyzing DNA are being applied in a wide range of fields, from agriculture to forensics to environmental science. Finally, and perhaps most importantly, the human genome raises profound ethical and societal questions. As we gain the ability to manipulate our genes, we must grapple with the ethical implications of genetic engineering, gene therapy, and genetic screening. It's a path with tremendous promise, but one that demands careful consideration of the potential consequences. In essence, the human genome is a treasure trove of information, a blueprint that holds the secrets to our past, present, and future. Unveiling its secrets is a journey that will continue to transform our understanding of ourselves and the world around us. It's a journey we're only just beginning. All right, let's dive into the very core of what makes you, well, you. We're talking about DNA, genes, and chromosomes, the fundamental building blocks of the human genome. Think of it as decoding the language of life itself. First up, DNA, or deoxyribonucleic acid. This is the molecule that carries all the genetic instructions for building and operating a human being, or any living organism for that matter. It's a double helix, a twisted ladder, an elegant structure that has captivated scientists for decades. Imagine two long strands winding around each other, connected by rungs. These rungs aren't made of wood, of course. They're formed by chemical bases. There are 4 types of these bases, and they go by the letters A, T, C, and G. A stands for adenine, T for thymine, C for cytosine, and G for guanine. Now, here's the clever part. A always pairs with T, and C always pairs with G. This complementary pairing is crucial. It means that if you know the sequence of bases on one strand of the DNA ladder, you automatically know the sequence on the other. This is how genetic information is stored and replicated with amazing accuracy. These of T, C, and G bases aren't just randomly arranged. Their specific sequence acts as a code, a set of instructions, much like the O South and 1 South that make up computer code. And the length of the sequences are staggering. The entire human genome is estimated to contain around 3 billion of these base pairs. That's a lot of information packed into something so tiny it exists inside of almost every cell in your body. Now, let's zoom in a bit more. Specific sequences of these bases make up what we call genes. Genes are the basic units of heredity. Think of them as individual recipes in a cookbook. Each gene contains the instructions for making a specific protein, and proteins are the workhorses of the cell. They carry out all sorts of functions, from building tissues and organs to regulating chemical reactions and fighting off infections. So, how does a gene actually make a protein? It's a 2-step process. First, the DNA sequence of a gene is transcribed into a molecule called RNA, or ribonucleic acid. RNA is similar to DNA, but it's single-stranded and has a slightly different sugar molecule. This RNA molecule acts as a messenger, carrying the genetic instructions from the nucleus, the cell's control center, to the ribosomes, the protein-making machinery located in the cytoplasm. At the ribosome, the RNA sequence is translated into a sequence of amino acids. Amino acids are the building blocks of proteins. There are 20 different amino acids, and their specific order determines the structure and function of the protein. Think of it like assembling Lego bricks in a certain order to build a specific model. Once the amino acid chain is complete, it folds into a unique 3-dimensional shape, which is essential for its function. The human genome contains an estimated 20,000 to 25,000 genes. That sounds like a lot, but it's actually surprisingly fewer than some simpler organisms. For example, A rice plant has more genes than a human. This highlights the fact that complexity isn't just about the number of genes, but also about how those genes are regulated and interact with each other. It's the intricate interplay of genes that gives rise to the incredible diversity of life. But where do we find all this DNA and these genes? They're organized into structures called chromosomes. Imagine taking all those long DNA strands and carefully winding them up into compact packages. That's essentially what chromosomes are. They're the organizational units that allow the massive amount of DNA to fit inside the cell's nucleus. Human cells have 23 pairs of chromosomes, for a total of 46. One set of 23 chromosomes comes from your mother, and the other set comes from your father. Each chromosome contains hundreds or even thousands of genes. These genes are arranged in a specific order along the chromosome, and their location is called a locus. Out of the 23 pairs, 22 are called autosomes. These chromosomes carry the genes for most of your traits, like eye color, height, and susceptibility to certain diseases. The remaining pair are the sex chromosomes, which determine your biological sex. Females have 2 X chromosomes, 20, while males have 1 X and 1 Y chromosome, XY. The Y chromosome contains the SRY gene, which is the master switch for male development. Chromosomes aren't just static structures. They play a critical role in cell division. When a cell divides, the chromosomes are duplicated, and each daughter cell receives a complete set. This ensures that all the cells in your body have the same genetic information. Now, let's talk about variations. While we all share the same basic set of genes, there are subtle differences in the DNA sequence that make each of us unique. These differences are called genetic variations or polymorphisms. They can be as small as a single base change, or they can involve larger stretches of DNA. These variations are what make us different from each other. They influence our physical traits, our susceptibility to diseases, and even our personality. Most genetic variations are harmless, but some can have significant effects on health. For example, some variations can increase the risk of developing cancer, heart disease, or Alzheimer's disease. Understanding the structure of DNA, the function of genes, and the organization of chromosomes is crucial for understanding how the human genome works. It's the foundation upon which all other genomic knowledge is built. By decoding the code of life, we can gain insights into our biology, our ancestry, and our health. And this is only the beginning. The field of genomics is constantly evolving, and we're learning new things about the human genome every day. All right, let's talk size. When we say the human genome is vast, we're not kidding. Imagine a book, then imagine a library. Then imagine every library in the world combined. That still wouldn't quite capture it. The human genome, in its entirety, contains approximately 3 billion DNA base pairs. Think of these base pairs, adenine, thymine, cytosine, and guanine, as the individual letters in a massive instruction manual, a blueprint for building and maintaining a human being. Now, if we were to print out this entire sequence, letter by letter, in a standard font size, it would fill over 200 volumes, each the size of a hefty phone book. That's a lot of reading material, and that's just one copy of your genome. Every single cell in your body, with a few exceptions like mature red blood cells, contains a complete copy of this incredible instruction set. It's like each cell has its own personal library of human design. But here's where it gets really interesting. Size isn't everything. It's the complexity of how this information is organized that truly sets the human genome apart. If we just had a linear sequence of 3 billion base pairs, it would be like having a string of letters with no spaces, punctuation, or formatting. Meaningless gibberish. The genome is anything but gibberish. It's a highly structured, intricately organized system with multiple layers of regulation and interaction. A crucial element of this organization comes in the form of genes. These are the functional units of heredity, the specific sequences of DNA that code for proteins. Proteins, as you probably know, are the workhorses of the cell, performing a vast array of functions from catalyzing biochemical reactions to building cellular structures. For a long time, scientists believed that a significant portion of the genome was dedicated to coding for proteins. But as the Human Genome Project unfolded, a surprising revelation emerged. Only about 1 to 2% of our DNA actually codes for proteins. This means that the vast majority of our genome, over 98%, is non-coding. For years, this non-coding DNA was dismissed as junk DNA, a leftover from our evolutionary past, serving no apparent purpose. However, research in recent years has revealed that this junk is anything but. In fact, it plays a crucial role in regulating gene expression, controlling when, where, and how much of a particular protein is produced. Think of it like this. The genes are the individual recipes in a cookbook, but the non-coding DNA is the editor, the index, and the table of contents. It determines which recipes are used, when they're used, and how they're modified to create the final dish. This regulatory role is essential for proper development and function, allowing different cell types in our body to express different sets of genes, even though they all contain the same underlying DNA. The complexity doesn't stop there. The human genome is also highly dynamic. It's not a static blueprint. but rather a constantly evolving and adapting system. Genes can be turned on and off in response to environmental cues, developmental signals, and even our own behavior. This dynamic regulation allows us to adapt to changing conditions and maintain homeostasis. Furthermore, the genome is not just a collection of individual genes acting in isolation. Genes interact with each other in complex networks, influencing each other's expression and function. These networks are essential for orchestrating complex biological processes, such as development, immunity, and metabolism. Understanding these gene networks is a major challenge in genomics research, but it's crucial for understanding how the genome functions as a whole, adding another layer of complexity. The genome is also subject to various modifications, such as DNA methylation and histone modification. These modifications don't change the underlying DNA sequence, but they can affect how genes are expressed. Think of them as sticky notes attached to the DNA, signaling which genes should be turned on or off. These epigenetic modifications can be influenced by environmental factors such as diet and exposure to toxins, and they can even be passed down from one generation to the next, adding another layer of inheritance beyond the DNA sequence itself. So, what does all of this mean? It means that the human genome is far more than just a collection of genes. It's a vast, intricate, and dynamic system with multiple layers of organization, regulation, and interaction. It's a complex ecosystem where genes, non-coding DNA, regulatory elements, and epigenetic modifications all work together to create and maintain a human being. And while the sheer size of the genome is impressive, it's the complexity of its organization and function that truly makes it remarkable. Unraveling this complexity is one of the greatest challenges in modern science, but it's a challenge that promises to revolutionize our understanding of human biology and disease. Because within those 3 billion base pairs lies the key to understanding who we are, where we come from, and what makes us unique. The next step is deciphering the secrets hidden within the non-coding majority. And that, as we'll see, is where some of the most exciting discoveries are being made. Okay, let's dive into the fascinating world of non-coding DNA. For a long time, scientists viewed large portions of our genome as junk DNA. This label stemmed from the fact that these regions don't directly code for proteins, the workhorses of our cells. But, as it turns out, dismissing them as mere evolutionary leftovers was a profound misunderstanding. Imagine the human genome as an incredibly complex instruction manual. The genes, the protein coding sequences, are like specific recipes for making particular tools. But a cookbook isn't just a list of recipes. is it? It has introductions, explanations of techniques, tables of contents, and an index. These elements guide you through the recipes and tell you how to use them most effectively. That's what non-coding DNA does. It's not directly making the proteins, but it's controlling how they're made, when they're made, and where they're made. So what exactly is this non-coding DNA doing? Well, it's a symphony of different elements, each with its own role. One of the most important functions is gene regulation. Think of gene regulation as the volume control for our genes. We don't need all of our genes turned on all the time in every cell. A muscle cell needs different proteins than a brain cell, and a developing embryo needs different proteins than an adult. Non-coding DNA contains sequences that act as switches, enhancers, and silencers, dictating whether a gene is active or inactive. These regulatory elements bind to proteins called transcription factors. Transcription factors are like specialized musicians that read the non-coding sequence and then either amplify or dampen the activity of a nearby gene. Some non-coding regions are promoters acting as landing pads where the transcription machinery assembles to initiate gene transcription. Others are enhancers, boosting gene expression from a distance. And still others are silencers, shutting genes down entirely. The location of these regulatory elements can be quite surprising. They might be located thousands or even millions of base pairs away from the genes they control. This means the DNA needs to fold into complex three-dimensional structures within the nucleus to bring these elements into physical proximity with their target genes. Imagine folding a long piece of string so that 2 points that are far apart on the string actually touch. This spatial organization of the genome is crucial for proper gene regulation. Another type of non-coding DNA includes elements that are transcribed into RNA but not translated into proteins. These are called non-coding RNAs, and they come in various flavors, each with unique functions. For example, ribosomal RNA, RNA, and transfer RNA, tRNA are essential for protein synthesis. They're like the tools and translators that allow the recipes to be followed accurately. But there are also other non-coding RNAs, like microRNAs, miRNAs, and long non-coding RNAs, LNC RNAs. MicroRNA molecules that regulate gene expression by binding to messenger RNA, mRNA, the intermediary molecule that carries the genetic code from DNA to the ribosomes. When a mRNA binds to an mRNA, it can either block its translation into protein or trigger its degradation. It's like a targeted sabotage of the genetic message, Long non-coding RNAs, on the other hand, are much longer and more diverse. They can act as scaffolds, bringing together different proteins to form complexes that regulate gene expression. They can also bind to DNA directly, influencing chromatin structure and accessibility. Chromatin is the complex of DNA and proteins that makes up our chromosomes. The structure of chromatin determines whether genes are accessible to the transcription machinery. Tightly packed chromatin generally silences genes, while loosely packed chromatin allows genes to be expressed. Long non-coding RNAs can influence this packaging, turning genes on or off. Then there are structural elements within the non-coding regions. These include centromeres, which are crucial for chromosome segregation during cell division, and telomeres, which protect the ends of our chromosomes from degradation. Without these structural elements, our genome would be unstable and our cells wouldn't be able to divide properly. One particularly fascinating category of non-coding DNA is transposable elements, also known as jumping genes. These are DNA sequences that can move from one location in the genome to another. They were initially viewed as selfish genetic elements, simply replicating themselves at the expense of the host genome. However, we now know that they can also play important roles in gene regulation and genome evolution. In fact, the insertion of a transposable element near a gene can alter its expression pattern, leading to new traits and adaptations over millions of years. Mutations can accumulate in these non-coding regions, leading to variations between individuals. These variations can affect gene regulation and contribute to differences in traits, including susceptibility to disease. In other words, even though these non-coding regions don't directly code for proteins, variations in these sequences can have profound effects on our health and well-being. Understanding the function of non-coding DNA is one of the biggest challenges in modern genomics. It's a complex and dynamic landscape, and we're only beginning to scratch the surface. But with each new discovery, we gain a deeper appreciation for the intricate workings of the human genome and the crucial role of these once dismissed junk sequences. They are the unsung heroes of our genetic code, orchestrating the symphony of life within our cells. The story of the non-coding genome is far from over, and its secrets are still being unveiled. As we continue to explore this vast and mysterious territory, we're sure to uncover even more surprising and important insights into the complexity of human biology. The story of understanding the human genome is, in many ways, the story of the Human Genome Project, a colossal undertaking, international in scope, and driven by a singular, audacious goal: to read, for the first time, the entire genetic instruction manual of a human being. To map the complete sequence of the human genome The seeds of this ambition were sown in the late 1980s. Scientists, armed with increasingly powerful tools for manipulating and analyzing DNA, began to envision the possibilities. Imagine, they thought, if we could decipher the entire sequence, all 3 billion base pairs that make up our genome, what secrets would it unlock? What diseases could we conquer? What insights into our own evolution could we gain? But the scale of the challenge was daunting. Sequencing even relatively small fragments of DNA was a laborious and time-consuming process. To tackle the entire human genome seemed almost impossibly ambitious. There was resistance, skepticism, concerns about the cost and the practicality of such an endeavor. Some argued that the resources could be better spent on more immediate, targeted research. However, the visionaries persisted. Prominent figures like Nobel laureate Walter Gilbert and James Watson, co-discoverer of the structure of DNA, championed the project. They argued persuasively that the potential benefits outweighed the risks, that the Human Genome Project was not just a scientific endeavor, but a fundamental investment in the future of human health and knowledge. The official launch came in 1990 with the backing of governments and research institutions around the world. The Human Genome Project was underway. The initial goals were clearly defined: to identify all the approximately 20,000 to 25,000 genes in human DNA, to determine the sequences of the 3 billion chemical base pairs that make up human DNA, to store this information in databases, to develop tools for data analysis, and to address the ethical, legal, and social issues arising from the project. The project adopted a multi-pronged approach. One key strategy involved breaking the genome down into smaller, more manageable fragments. These fragments were then cloned, creating multiple identical copies, allowing scientists to work with sufficient quantities of DNA. These cloned fragments were then mapped to specific locations on chromosomes. Think of it like creating a giant jigsaw puzzle where each piece represents a fragment of DNA. And the challenge is to fit all the pieces together in the correct order. Another crucial aspect of the project was the development of new sequencing technologies. Existing methods were simply too slow and inefficient to handle the sheer volume of DNA that needed to be analyzed. Researchers pushed the boundaries of automation and miniaturization, creating machines that could rapidly sequence DNA fragments with increasing accuracy. These technological breakthroughs were essential for accelerating the pace of the project. Early on, the Human Genome Project faced challenges, as any endeavor of this scale would. There were debates about the best sequencing strategies, the management of data, and the allocation of resources. There were also ethical concerns about the potential misuse of genetic information. Would this knowledge lead to genetic discrimination? Would it exacerbate existing inequalities in healthcare? These were important questions that needed to be addressed. Despite these challenges, the project made steady progress. Year after year, scientists chipped away at the genome, sequencing fragment after fragment, refining their techniques, and building ever more comprehensive maps. Initial findings started trickling out, offering tantalizing glimpses into the organization and function of our genetic code. Regions of the genome associated with certain diseases began to be identified. New insights into the evolution of humans and our relationship to other species emerged. Then, in 2001, came a landmark achievement. The Human Genome Project announced the publication of a working draft of the human genome sequence. It wasn't perfect. There were still gaps and ambiguities, but it was a monumental milestone, providing a comprehensive, albeit incomplete, blueprint of our genetic makeup. This working draft represented a huge leap forward in our understanding of human biology. The initial findings were both surprising and humbling. One of the biggest surprises was the relatively small number of genes. Scientists had initially estimated that humans might have as many as 100,000 genes. The working draft revealed that the actual number was closer to 0 to 25,000. This raised profound questions about the complexity of human biology. If we didn't have significantly more genes than simpler organisms, what made us so different? The answer, it turned out, lies not just in the number of genes, but in how those genes are regulated and expressed. The Human Genome Project revealed that a large portion of our genome, often referred to as non-coding DNA, plays a critical role in controlling gene activity. These non-coding regions contain regulatory elements that switch genes on and off, fine-tuning their expression in different tissues and at different times. It's this complex interplay between genes and their regulatory elements that ultimately determines our development, physiology, susceptibility to disease. The initial findings also highlighted the remarkable similarity between the genomes of all human beings. While there is certainly genetic variation between individuals, the vast majority of our DNA is identical. This underscores our shared ancestry and the fundamental unity of the human species. The differences that do exist, however, are incredibly important. They contribute to our individual traits, our susceptibility to certain diseases, and our responses to medications. Understanding these genetic variations is key to unlocking the potential of personalized medicine. Another key finding from the Human Genome Project was the discovery of repetitive sequences within our DNA. These repetitive sequences make up a significant portion of our genome and were initially dismissed as junk DNA. However, Scientists now recognize that these repetitive sequences play important roles in genome organization and stability. Some repetitive sequences, for example, help to protect the ends of our chromosomes, preventing them from being damaged. Others contribute to the formation of specialized structures within the nucleus of our cells. The Human Genome Project's working draft was a monumental achievement, but the work was far from over. The remaining gaps needed to be filled, the errors needed to be corrected, and the entire sequence needed to be refined. It was a task akin to polishing a rough diamond, gradually revealing its brilliance. The final, complete sequence of the human genome was published in 2003, marking the official completion of the Human Genome Project, an endeavor that transformed our understanding of life itself. The quest to decipher the human genome was for many years an exercise in painstaking, almost unimaginable effort. Imagine trying to read a book billions of letters long without knowing where the words begin or end, and with a technology that could only read a few letters at a time. The early methods of DNA sequencing were slow, expensive, and required a huge amount of manual labor. But the story of genome analysis is ultimately one of innovation, of brilliant minds relentlessly pursuing faster, cheaper, and more accurate ways to unlock the secrets of our genetic code. The first real breakthrough came in the mid-1970s with the development of what's now known as Sanger sequencing, named after its inventor, Frederick Sanger. This method, sometimes referred to as chain termination sequencing, relied on the use of modified nucleotides called deoxynucleodes. These special nucleotides, when incorporated into a growing DNA strand, would halt the replication process. By labeling these deoxynucleotides with different fluorescent dyes, scientists could generate a series of DNA fragments of varying lengths, each ending with a specific base: adenine, guanine, cytosine, or thymine. Think of it like making copies of a recipe, but each copy gets cut off at a different ingredient. By sorting the copies by size and seeing what ingredient is last on each one, you can reconstruct the entire recipe. These fragments were then separated by size using gel electrophoresis, a process where DNA molecules are pulled through a gel-like substance by an electric field. Smaller fragments move faster, allowing for separation based on size. By reading the fluorescent labels on each fragment as they passed a detector, scientists could determine the order of the bases, effectively reading the DNA sequence. Sanger sequencing was revolutionary. It was the method used to sequence the first complete viral genome and, crucially, became the workhorse of the Human Genome Project. However, it was still relatively slow and laborious. Each DNA fragment that could be sequenced in a single reaction, a read, was only a few hundred base pairs long. This meant that to sequence the entire human genome, which is over 3 billion base pairs, the DNA had to be broken into millions of smaller fragments, sequenced individually, and then painstakingly pieced back together. This process, called shotgun sequencing, was like trying to assemble a giant jigsaw puzzle with billions of pieces, and where you don't even have the picture on the box. The Human Genome Project spurred further innovation in sequencing technology. Capillary electrophoresis, an improvement on gel electrophoresis, automated the process and allowed for higher throughput. Instead of running samples in gels, the DNA fragments were separated in thin glass capillaries, allowing for faster separation and more efficient detection. This advancement significantly increased the speed and reduced the cost of Sanger sequencing, making it a more practical tool for large-scale genome sequencing projects. But the real game changer arrived in the mid-2000s: next-generation sequencing, or NGS. These technologies, also known as massively parallel sequencing, completely transformed the field by allowing millions or even billions of DNA fragments to be sequenced simultaneously. Unlike Sanger sequencing, which reads each fragment individually, NGS platforms sequence DNA fragments in parallel, greatly increasing the throughput and reducing the cost per base. Several different NGS technologies emerged, each with its own advantages and disadvantages. One of the most widely used is sequencing by synthesis, developed by Illumina. This method involves attaching millions of DNA fragments to a solid surface, amplifying them to create clusters of identical DNA molecules, and then sequencing them simultaneously by adding fluorescently labeled nucleotides, one at a time. As each nucleotide is added, a camera captures the fluorescent signal, allowing the sequence to be determined. Another popular NGS technology is purist sequencing, developed by 454 Life Sciences. This method detects the release of pyrophosphate, a byproduct of DNA synthesis, as each nucleotide is incorporated into a growing DNA strand. The pyrophosphate is converted into light, which is then detected by a sensor. These NGS technologies drastically reduced the cost and increased the speed of genome sequencing. What once cost hundreds of millions of dollars and took years to accomplish could now be done for a few $1,000s in a matter of days. This opened up a vast array of new possibilities, from personalized medicine to large-scale population genetics studies. But the revolution didn't stop there. The quest for even faster, cheaper, and more accurate sequencing technologies continues. Third-generation sequencing technologies, such as those developed by Pacific Biosciences, PacBio, and Oxford Nanopore Technologies offer the ability to sequence single molecules of DNA in real time without the need for amplification. PacBio sequencing uses a technique called single molecule real-time, or SMRT sequencing. This method involves attaching a DNA polymerase to the bottom of a tiny well called a zero-mode waveguide, and then monitoring the incorporation of fluorescently labeled nucleotides as the polymerase synthesizes a new DNA strand. Because the sequencing occurs in real time, PacBio sequencing can generate very long reads, often tens of thousands of base pairs long. These long reads are particularly useful for sequencing complex genomes, such as those containing repetitive sequences or structural variations. Oxford Nanopore sequencing uses a different approach. This method involves passing a single strand of DNA through a tiny protein nanopore embedded in a membrane. As the DNA molecule passes through the pore, it disrupts an electrical current. By measuring the changes in current, the sequence of the DNA can be determined. Oxford Nanopore sequencing is particularly attractive because it is relatively inexpensive and portable. The sequencing devices are small enough to fit in the palm of your hand, making them ideal for field applications. The impact of these sequencing breakthroughs has been profound. We have gone from struggling to sequence a few thousand base pairs to being able to sequence entire human genomes in a matter of hours. This has accelerated our understanding of genetics, disease, and evolution, and has paved the way for personalized medicine and other transformative applications. The ongoing development of new sequencing technologies promises to further revolutionize the field, bringing us closer to unlocking the full potential of the human genome. Personalized medicine. It's a phrase that's been tossed around a lot lately, and for good reason. It represents a paradigm shift in healthcare, moving away from a one-size-fits-all approach to one that is tailored to the individual. And at the heart of this revolution lies genome sequencing. Think of it like this. For centuries, doctors have been treating symptoms, often without fully understanding the underlying cause. It's like trying to fix a car engine without knowing how it works. Genome sequencing, however, provides us with the instruction manual, the complete blueprint of our bodies. It allows us to understand the unique genetic variations that make each of us different, and crucially, how those variations influence our health and our response to treatment. The journey to widespread genome sequencing began with the Human Genome Project, a monumental effort to map the entire human genome. While the project provided a reference genome, a standard, it also highlighted the immense variability that exists between individuals. This is where personalized medicine truly begins. So, how is genome sequencing transforming healthcare? Let's start with diagnosis. For many diseases, especially rare genetic disorders, diagnosis can be a long and frustrating process, often involving multiple tests and specialist consultations. Genome sequencing can significantly shorten this diagnostic odyssey. By analyzing a patient's DNA, doctors can often pinpoint the exact genetic mutation causing the condition, leading to a faster and more accurate diagnosis. This is particularly crucial for conditions where early intervention is key to improving outcomes. But the benefits extend far beyond rare diseases. Genome sequencing is also proving invaluable in understanding and treating more common conditions, such as cancer, heart disease, and diabetes. In cancer, for example, sequencing the tumor's genome can reveal specific mutations that are driving its growth. This information can then be used to select the most effective targeted therapies, drugs that specifically attack the cancer cells while sparing healthy tissue. It's about understanding the enemy at a molecular level and deploying the right weapons. Consider pharmacogenomics, the study of how genes affect a person's response to drugs. We all metabolize medications differently. What works well for one person might have no effect or even cause adverse reactions in another. Genome sequencing can help predict how a patient will respond to a particular drug, allowing doctors to choose the right medication and dosage from the outset. This not only improves treatment outcomes, but also reduces the risk of side effects and wasted healthcare resources. Imagine a future where you receive a medication specifically tailored to your genetic makeup, maximizing its effectiveness and minimizing potential harm. That future is closer than you think. Another exciting area is preventative medicine. Genome sequencing can identify individuals who are at increased risk of developing certain diseases, such as Alzheimer's, Parkinson's, or specific types of cancer. While knowing your genetic predisposition can be daunting, it also empowers you to take proactive steps to reduce your risk. This might involve lifestyle changes, such as adopting a healthier diet and exercise routine, or undergoing regular screenings to detect the disease at an early, more treatable stage. For better health, but the promise of personalized medicine extends beyond treatment and prevention. It also holds the potential to improve our understanding of disease biology. By studying the genomes of large populations, researchers can identify novel genes and pathways involved in disease development. This, in turn, can lead to the development of new and more effective therapies. It's about using the power of genomics to unravel the complexities of human disease and find innovative ways to combat them. The applications of genome sequencing are vast and rapidly expanding. From predicting drug response to identifying disease risks and developing targeted therapies, it's transforming virtually every aspect of healthcare. And as the cost of sequencing continues to fall and the technology becomes more accessible, personalized medicine is poised to become the standard of care. Of course, there are challenges to overcome. Interpreting genomic data requires sophisticated analytical tools and expertise. We need to develop better algorithms and databases to translate genetic information into clinically meaningful insights. We also need to address ethical concerns related to genetic privacy and the potential for discrimination. Ensuring that genetic information is used responsibly and ethically is paramount. Despite these challenges, the potential benefits of personalized medicine are undeniable. It represents a fundamental shift towards a more precise, proactive, and patient-centered approach to healthcare. It's about empowering individuals to take control of their health and working with their doctors to develop personalized treatment plans based on their unique genetic makeup. As we continue to unlock the secrets of the human genome, we are paving the way for a future where healthcare is truly personalized, leading to healthier and longer lives for all. Genetic predisposition. It's a phrase we hear often, but what does it truly mean? In essence, it refers to an increased likelihood of developing a particular disease based on an individual's genetic makeup. It's not a guarantee, but rather a heightened risk compared to someone without those specific genetic variants. Think of it as holding a hand of cards. Some cards make you more likely to win, but you still need to play the game. Our genomes are incredibly complex, and within them lie countless variations or polymorphisms. These variations, often subtle changes in the DNA sequence, are what make each of us unique. Some of these variations, however, are linked to an increased risk of certain diseases. Identifying these genes and their associated variations is a monumental task, but one that holds immense potential for preventative medicine. How do scientists go about uncovering these genetic links? Well, one of the most common approaches is through genome-wide association studies, or GWAS. These studies involve scanning the entire genomes of large groups of people, both those with a particular disease and those without. Researchers then look for genetic markers, specific DNA sequences that are more frequently found in individuals with the disease. If a marker is consistently associated with a disease across multiple studies, it suggests that a gene near that marker may play a role in its development. Imagine comparing millions of books, each with slight variations in spelling and punctuation. GWAs are like trying to identify which specific typos are most common in books written by authors who later developed a specific writing style. It's a massive undertaking, requiring powerful computers and sophisticated statistical analysis. But GWAs are not without their limitations. They primarily identify common genetic variations, meaning those present in at least 1% of the population. This means that rare genetic variants, which can also significantly contribute to disease risk, may be missed. Additionally, GWAs often identify regions of the genome associated with disease, rather than pinpointing the exact causative gene. Further research is then needed to identify the specific gene within that region and understand how it contributes to disease development. Another approach involves studying families with a history of a particular disease. In these cases, researchers can trace the inheritance of the disease through generations, looking for genetic markers that are consistently passed down along with the disease. This method is particularly useful for identifying genes involved in rare inherited disorders. These studies allow researchers to analyze which sections of the genome are shared between all affected family members. This approach is like trying to identify key inherited artifacts that may be connected to the disease across different branches of the same family. Once a gene is identified as being associated with a disease, the next step is to understand how that gene contributes to the disease process. This often involves studying the function of the gene and how its activity is altered by the disease-associated variations. For example, a variation in a gene might affect the production of a protein, or it might alter the way the protein functions. These changes can then lead to disruptions in cellular processes, ultimately contributing to disease development. Understanding the functional role of these genes is crucial for developing targeted therapies. If we know exactly how a gene contributes to a disease, we can design drugs or other interventions that specifically target that gene or its protein product. This approach is known as personalized medicine, and it holds immense promise for treating diseases more effectively and with fewer side effects. Genetic predisposition is relevant to a wide range of diseases and health risks, from common conditions like heart disease, diabetes, and certain cancers, to rarer inherited disorders like cystic fibrosis and Huntington's disease. Knowing your genetic predispositions can empower you to take proactive steps to reduce your risk of developing certain diseases. For example, if you know you have a genetic predisposition to heart disease, you might choose to adopt a healthier lifestyle, including a balanced diet, regular exercise, and avoiding smoking. Similarly, if you have a genetic predisposition to certain types of cancer, you might opt for more frequent screenings to detect the disease early when it's more treatable. Genetic testing is becoming increasingly accessible, allowing individuals to learn about their genetic predispositions for a variety of diseases. However, it's important to remember that genetic testing is just one piece of the puzzle. Your genes are not your destiny. Environmental factors, lifestyle choices, and other factors also play a significant role in determining your overall health. It's a complex interplay, nature and nurture working together. Moreover, it's crucial to ensure that genetic information is used responsibly and does not lead to discrimination or other forms of prejudice. Protecting genetic privacy is paramount, and regulations are needed to prevent misuse of genetic data. Insurance companies and employers should not be able to discriminate against individuals based on their genetic predispositions. Finally, it's important to remember that our understanding of the human genome is constantly evolving. New genes and genetic variations are being discovered all the time, and our knowledge of how genes interact with each other and with the environment is constantly expanding. The journey of unlocking the secrets of the human genome is far from over. As we continue to unravel the complexities of our genetic code, we can expect to see even greater advances in personalized medicine and preventative healthcare. All right, let's dive into pharmacogenomics, tailoring drug treatments based on individual genetic profiles. Imagine a world where medication is prescribed not just based on your symptoms, but on your unique genetic makeup. That world is closer than you think, thanks to the burgeoning field of PHRMACO genomics at its core. Pharmacogenomics is the study of how genes affect a person's response to drugs. It's the bridge between genomics and pharmacology, aiming to optimize drug therapy by ensuring patients receive the right drug at the right dose at the right time. This personalized approach is a departure from the traditional one-size-fits-all model, which often leads to ineffective treatments or WORSE, adverse drug reactions. So, how does your genome influence your reaction to medication? Well, several factors come into play, all rooted in your DNA. One crucial aspect is drug metabolism. Many medications are broken down by enzymes in the body, primarily in the liver. The genes that code for these enzymes can vary significantly from person to person. These variations, known as genetic polymorphisms, can affect how quickly or slowly someone metabolizes a drug. For example, consider the CYP2D6 gene. This gene encodes an enzyme responsible for metabolizing a wide range of commonly prescribed medications, including antidepressants, pain relievers, and beta blockers. Some individuals have genetic variations that make them poor metabolizers, meaning they break down these drugs very slowly. As a result, standard doses can lead to a buildup of the drug in their system, increasing the risk of side effects or toxicity. Conversely, others are ultra-rapid metabolizers, breaking down drugs so quickly that standard doses become ineffective. They might not experience the intended therapeutic benefit, leading to frustration and potentially unnecessary dose escalations. Pharmacogenomic testing can identify these individuals, allowing doctors to adjust dosages accordingly or choose alternative medications metabolized by different pathways. Another important genetic influence lies in drug transporters. These proteins act like gatekeepers, shuttling drugs into and out of cells. Genetic variations in transporter genes can affect how well a drug reaches its target, influencing its effectiveness. For instance, some transporters might pump a drug out of a cancer cell, reducing its ability to kill the tumor. Identifying these variations can help oncologists select drugs that are more likely to be effective in a given patient. But it's not just about metabolism and transport. Your genes can also influence drug targets themselves. Many drugs work by binding to specific proteins in the body, like receptors or enzymes. Genetic variations can alter the structure of these targets, affecting how well a drug binds and, consequently, how effective it is. Think of it like a lock and key. If the lock, the target protein, is slightly different, the key, the drug, might not fit as well. A prime example is the gene VKORC1, which encodes a protein involved in vitamin K metabolism. This protein is the target of warfarin, a commonly prescribed blood thinner. Genetic variations in VKORC1 affect how sensitive individuals are to warfarin. Some people require much lower doses to achieve the desired blood thinning effect, while others need significantly higher doses. Pharmacogenomic testing for VKORC1 can help doctors determine the optimal starting dose of warfarin, reducing the risk of bleeding complications or blood clots. The applications of pharmacogenomics are vast and expanding. It's already being used in several areas of medicine, including oncology, cardiology, psychiatry, and pain management. In oncology, for example, pharmacogenomic testing can help predict a patient's response to chemotherapy drugs, allowing doctors to choose the most effective treatment regimen while minimizing side effects. In cardiology, it can guide the selection of antiplatelet medications after a heart attack or stent placement, reducing the risk of blood clots. In psychiatry, it can help optimize antidepressant therapy, increasing the chances of finding a medication that works effectively with minimal side effects. And in pain management, it can help predict a patient's response to opioid pain relievers, reducing the risk of addiction and overdose. Despite its promise, pharmacogenomics faces several challenges. One major hurdle is the cost of testing. While the price of genetic sequencing has fallen dramatically in recent years, pharmacogenomic tests can still be expensive, limiting their accessibility. Another challenge is the lack of widespread awareness and education among healthcare providers. Many doctors are not yet familiar with pharmacogenomics and its potential benefits, hindering its adoption into routine clinical practice. Furthermore, the interpretation of pharmacogenomic test results can be complex. Genes often interact with each other and with environmental factors, making it difficult to predict drug response with certainty. More research is needed to fully understand the interplay between genes, drugs, and other variables. However, the future of pharmacogenomics is bright. As technology advances and costs continue to decline, genetic testing will become more accessible and affordable. Increased awareness and education among healthcare providers will pave the way for wider adoption. And as we gain a deeper understanding of the complex interactions between genes and drugs, we'll be able to develop more precise and personalized treatment strategies. Imagine a future where every patient receives a comprehensive genetic profile at birth, providing a roadmap for optimizing their healthcare throughout their life. This roadmap would include information on their predicted drug responses, allowing doctors to prescribe medications tailored to their individual needs. This is the ultimate goal of pharmacogenomics: to transform medicine from a reactive, trial-lander approach to a proactive, personalized one, leading to better health outcomes for everyone. It's not science fiction. It's the direction medicine is headed. One gene at a time. Gene therapy. The very phrase evokes images of a future where inherited diseases are relics of the past. It's a field brimming with potential, a beacon of hope for families grappling with conditions passed down through generations. But what exactly is gene therapy, and how does it work? At its core, gene therapy aims to treat or even cure diseases by modifying a person's genes. This can involve several strategies. We might be talking about replacing a mutated gene that causes disease with a healthy copy, or perhaps inactivating a mutated gene that is functioning improperly. Another avenue is introducing a new gene into the body to help fight a disease. Think of it as rewriting the body's instruction manual to correct errors and restore proper function. Now, how do we actually deliver these therapeutic genes into the patient's cells? This is where vectors come into play. A vector is essentially a delivery vehicle, often a modified virus. Viruses have a natural ability to infect cells, and scientists have cleverly repurposed this ability to carry genetic material directly into the patient's cells. Why viruses? Well, they are highly efficient at entering cells. But don't worry, these viruses are engineered to be harmless. The disease-causing genes are removed, and the therapeutic gene is inserted in their place. It's like using a postal service that's been thoroughly vetted to ensure it only delivers the intended package. Several types of viral vectors are commonly used, each with its own advantages and disadvantages. Adenoviruses, for example, are known for their ability to infect a wide range of cells, but they can sometimes trigger an immune response. Adeno-associated viruses, or AAVs, are generally considered safer and less likely to provoke an immune reaction, but they can carry smaller genes. Retroviruses, including lentiviruses, can integrate their genetic material into the host cell's DNA, leading to long-term expression of the therapeutic gene. But they also carry a risk of disrupting other important genes. The choice of vector depends on several factors, including the type of cells that need to be targeted, the size of the therapeutic gene, and the desired duration of gene expression. It's a delicate balancing act, weighing the benefits of each vector against its potential risks. But viral vectors aren't the only option. Non-viral methods are also being developed, such as injecting DNA directly into the body or using lipid nanoparticles to encapsulate and deliver the therapeutic gene. These methods are generally considered safer than viral vectors, but they are often less efficient at delivering genes into cells. Once the vector delivers the therapeutic gene into the patient's cells, what happens next? Ideally, the gene will be expressed, meaning the cell will use the gene's instructions to produce a protein that can correct the underlying defect. This protein might replace a missing enzyme, block the activity of a harmful protein, or stimulate the immune system to fight cancer cells. Gene therapy can be broadly classified into two main types: somatic gene therapy and germline gene therapy. Somatic gene therapy targets non-reproductive cells, meaning that the genetic changes are not passed on to future generations. This is the most common type of gene therapy, and it's the focus of most current research. Germline gene therapy, on the other hand, targets reproductive cells, such as eggs or sperm, meaning that the genetic changes would be inherited by future generations. Germline gene therapy raises significant ethical concerns, and it is not currently being used in humans. The potential applications of gene therapy are vast. It's being explored as a treatment for a wide range of inherited diseases, including cystic fibrosis, spinal muscular atrophy, hemophilia, and sickle cell anemia. It also holds promise for treating cancer, infectious diseases, and even age-related conditions. One of the most notable success stories of gene therapy is the treatment of spinal muscular atrophy, or SMA. SMA is a devastating genetic disorder that causes muscle weakness and atrophy. A gene therapy called Zolgensma has been shown to dramatically improve the lives of children with SMA, allowing them to sit crawl, and even walk. This is a truly remarkable achievement, and it highlights the transformative potential of gene therapy. However, gene therapy is not without its challenges. One of the biggest hurdles is ensuring that the therapeutic gene is delivered to the right cells and expressed at the right level. Off-target effects, where the vector delivers the gene to the wrong cells, can lead to unintended consequences. Another challenge is the potential for immune responses to the vector or the therapeutic gene. Moreover, the long-term effects of gene therapy are not yet fully understood. While some gene therapies have shown durable benefits, others have seen the effects wane over time. More research is needed to determine how long gene therapies last and whether repeat treatments are necessary. The cost of some gene therapies cost hundreds of thousands or even millions of dollars per treatment, making them inaccessible to many patients. Efforts are underway to reduce the cost of gene therapy and make it more affordable. Despite these challenges, gene therapy is a rapidly evolving field with tremendous promise. As scientists continue to develop new and improved vectors, refine targeting strategies, and gain a better understanding of the long-term effects, gene therapy is poised to revolutionize the treatment of many diseases. It's a journey of continuous discovery, driven by the unwavering hope of a healthier future for all. The potential rewards are simply too great to ignore. The human genome, a marvel of biological engineering, holds within it the secrets to our very being. But with this power comes profound responsibility. We've journeyed far in understanding this intricate code, but now we must confront the complex ethical landscape surrounding genetic information and its applications. It's a landscape fraught with difficult questions, requiring careful consideration and open dialogue. At the heart of the matter lies the inherent tension between scientific progress and societal values. On one hand, we have the incredible potential to alleviate suffering, cure diseases, and even enhance human capabilities. On the other, we face the risks of discrimination, privacy violations, and unintended consequences that could reshape our society in unforeseen and potentially harmful ways. One of the most pressing ethical concerns revolves around genetic testing and screening. While the ability to identify genetic predispositions to diseases offers the hope of early detection and preventative measures, it also raises the specter of genetic discrimination. Imagine a world where individuals are denied employment or insurance coverage based solely on their genetic profile. A world where our genes predetermine our fate in the eyes of institutions. This is not a far-fetched scenario. Laws like the Genetic Information Non-Discrimination Act, or GINA in the United States, aim to prevent this type of discrimination. But the need for vigilance and stronger protections remains. Beyond discrimination, The widespread availability of genetic testing raises questions about privacy and data security. Who has access to your genetic information? How is it stored and protected? Could it be used against you without your knowledge or consent? The answers to these questions are far from straightforward. As genetic databases grow larger and more interconnected, the risk of data breaches and misuse increases. We must ensure that robust safeguards are in place to protect the privacy and confidentiality of genetic information. Recognizing that it is perhaps the most personal information we possess, another ethical dilemma arises in the realm of reproductive technologies. Pre-implantation genetic diagnosis, or PGD, allows prospective parents to screen embryos for genetic disorders before implantation. While PGD can prevent the birth of children with serious illnesses, it also opens the door to the selection of embryos based on other traits, such as sex or even physical characteristics. Where do we draw the line between preventing disease and designing designer babies? This is a question with no easy answers, and it demands careful consideration of the values we wish to uphold as a society. The implications of gene therapy, the process of altering a person's genes to treat or prevent disease, also require scrutiny. While gene therapy holds immense promise for curing inherited disorders like cystic fibrosis or sickle cell anemia, it also raises concerns about safety, efficacy, and accessibility. Are the risks of gene therapy outweighed by its potential benefits? How can we ensure that gene therapy is available to all who need it, not just the wealthy elite? And what about the potential for germline gene therapy, which would alter the genes of future generations? This is arguably the most controversial area of genetic engineering, as it raises the prospect of fundamentally changing the human species. The debate over germline gene editing is particularly fraught with ethical challenges. Proponents argue that it could eradicate inherited diseases and improve human health, while opponents warn of unintended consequences and the potential for unforeseen harms to future generations. The long-term effects of altering the human germline are unknown, and some fear that it could lead to a slippery slope toward eugenics. The discredited practice of selectively breeding humans to improve the species. The potential benefits must be weighed against the significant ethical risks. Furthermore, the increasing accessibility of genetic information raises complex issues surrounding informed consent. Do individuals fully understand the implications of genetic testing before they undergo it? Are they prepared to receive potentially life-altering information about their health risks? Ensuring that individuals have access to accurate and unbiased information about genetic testing is crucial for empowering them to make informed decisions about their healthcare. Genetic counseling plays a vital role in this process, helping individuals understand the complexities of genetic information and its potential impact on their lives. It's not just about the individual either. The potential for group harm also warrants careful consideration. Genetic research that focuses on specific populations, for instance, must be conducted in a way that respects their cultural values and avoids perpetuating harmful stereotypes. Historical injustices, such as the forced sterilization of marginalized groups, underscore the need for caution and ethical sensitivity in genetic research. As we delve deeper into the secrets of the human genome, we must also address the issue of intellectual property rights. Who owns the rights to genetic information? Should companies be allowed to patent genes or genetic technologies? These are complex legal and ethical questions with significant implications for access to genetic testing and treatments. Striking a balance between incentivizing innovation and ensuring equitable access to these life-saving technologies is essential. Ultimately, navigating the ethical landscape of genetic information requires a multidisciplinary approach involving scientists, ethicists, policymakers, and the public. Open dialogue and public engagement are crucial for fostering a shared understanding of the ethical implications of genetic technologies and for developing policies that promote responsible innovation. We must recognize that the ethical challenges posed by the human genome are not merely technical or scientific, but deeply human. They touch upon our fundamental values, our sense of identity, and our vision for the future. As we continue to unlock the secrets of the human genome, we must ensure that our scientific progress is guided by ethical principles, respect for human dignity, and a commitment to a just and equitable society. The power to decode and manipulate the human genome is a tremendous gift, but also a profound responsibility. How we choose to wield that power will determine the future of our species. Genetic privacy. It's a phrase we hear increasingly often, but what does it really mean? And why is it so critical in our increasingly genomic world? Simply put, genetic privacy is the right of individuals to control access to and use of their genetic information. It's about preventing the misuse of this data and guarding against potential discrimination based on our unique genetic blueprints. Think of your genome as a highly detailed instruction manual for you. It contains incredibly personal information, predisposing you to certain diseases, revealing ancestry, and even hinting at personality traits. Now imagine that manual falling into the wrong hands. The potential consequences are far-reaching and potentially devastating. One of the most pressing concerns is genetic discrimination. This can manifest in various forms, most commonly in employment and insurance. Imagine applying for a job, and the employer gains access to your genetic data. If that data reveals a predisposition to a latent set disease, they might unfairly deem you a less desirable candidate, fearing future healthcare costs or reduced productivity. Similarly, health insurance companies might use genetic information to deny coverage, raise premiums, or impose limitations based on perceived health risks. This isn't just hypothetical. There have been documented cases and anxieties surrounding these issues, even with legal protections in place. In the US, the Genetic Information Non-Discrimination Act, or GINA, offers some protection against genetic discrimination in employment and health insurance. GINA prohibits employers and health insurers from using genetic information to make decisions about hiring, firing, promotion, or coverage. However, GINA has its limitations. It doesn't cover life insurance, disability insurance, or long-term care insurance. This leaves individuals vulnerable to discrimination in these crucial areas. And even within its scope, proving discrimination based on genetic information can be challenging, requiring substantial evidence and legal expertise. Beyond employment and insurance, genetic privacy extends to other sensitive areas. Law enforcement agencies are increasingly using genetic genealogy databases to identify suspects in criminal investigations. While this can be a powerful tool for solving crimes, it also raises serious concerns about privacy and potential for misuse. These databases, often populated with genetic information voluntarily submitted by individuals for ancestry research, were never intended for law enforcement purposes. The practice of searching these databases for familial matches can cast a wide net, potentially implicating innocent individuals and violating their privacy rights. Direct-to-consumer genetic testing companies like 23andMe and Ancestry DNA have revolutionized access to genetic information. For a relatively low cost, individuals can gain insights into their ancestry, health, predispositions, and traits. However, this convenience comes with inherent privacy risks. These companies collect, store, and analyze vast amounts of genetic data, making them attractive targets for hackers and data breaches. Even if these companies have robust security measures in place, there's always a risk of unauthorized access or data leaks. Furthermore, the privacy policies of these companies can be complex and difficult to understand, leaving consumers unsure about how their data is being used and shared. Some companies may share anonymized or aggregated genetic data with third-party researchers or pharmaceutical companies, which raises concerns about potential commercial exploitation of personal genetic information. Another significant challenge to genetic privacy is the increasing availability of genetic data in research databases. While these databases are essential for advancing scientific knowledge and developing new treatments, they also pose privacy risks. Researchers often need access to identifiable genetic data to conduct meaningful analysis. However, ensuring the confidentiality and security of this data is crucial to protect the privacy of research participants. Data anonymization techniques, such as removing personal identifiers, can help to reduce the risk of re-identification. But these methods aren't foolproof, and advanced techniques, like differential privacy, are needed to provide stronger privacy guarantees. So, what can we do to protect our genetic privacy? Firstly, it's essential to be informed about the risks and potential consequences of sharing our genetic information. We should carefully review the privacy policies of direct-to-consumer genetic testing companies and understand how our data will be used. We should also be aware of our legal rights and protections under laws like GINA. Secondly, we need stronger legal and regulatory frameworks to protect genetic privacy. This includes extending GIN's protections to life insurance, disability insurance, and long-term care insurance. It also requires developing clear guidelines for law enforcement use of genetic genealogy databases and ensuring transparency and accountability in the collection, storage, and use of genetic data by research institutions and commercial entities. Thirdly, we need to promote the development and adoption of privacy-enhancing technologies such as secure multi-party computation and federated learning. These technologies allow researchers to analyze genetic data without directly accessing or sharing sensitive information. This can help to advance scientific knowledge while preserving individual privacy. Finally, we need to foster a culture of responsible data sharing and ethical conduct in the genomics community. This includes educating researchers, healthcare professionals, and the public about the importance of genetic privacy and promoting best practices for data security and confidentiality. Genetic privacy is not just a personal issue, it's a societal imperative. As our understanding of the human genome grows, and as genetic technologies become more widespread, protecting genetic privacy will become increasingly critical to ensure fairness, equality, and individual autonomy. The choices we make today about genetic privacy will shape the future of healthcare, research, and society as a whole. It's a conversation we must have and a challenge we must meet head-on. Genetic engineering. The very term conjures images of futuristic labs, ethical dilemmas, and the potential for a world irrevocably altered. And nowhere is this more potent, more laden with possibility and risk than when we consider the human germline. What exactly is the germline? Simply put, it's the line of cells, sperm and egg cells, that pass genetic information from one generation to the next. Changes made to these cells aren't confined to a single individual. They are heritable, meaning they are passed down to all subsequent descendants. This is in stark contrast to somatic cell therapy, which targets non-reproductive cells and only affects the individual receiving the treatment. The prospect of modifying the human germline is both tantalizing and terrifying. On the one hand, it offers the potential to eradicate inherited diseases, to eliminate genetic predispositions to cancer, Alzheimer's, or countless other debilitating conditions. Imagine a world where cystic fibrosis, Huntington's disease, and sickle cell anemia are relegated to the history books. A world where parents can ensure their children are born free from the genetic burdens that have plagued humanity for generations. This is the alluring promise of germline editing. It's the chance to rewrite the very blueprint of our species, to sculpt a healthier, more resilient future. By correcting disease-causing mutations in the germline, we could permanently remove them from the gene pool, preventing their transmission to future generations. But with such immense power comes immense responsibility. The potential benefits of germline editing are undeniable, but the risks, both known and unknown, are equally profound. One of the most pressing concerns is the possibility of unintended consequences. Our understanding of the genome, while vastly improved, is still far from complete. Genes don't operate in isolation. They interact in complex and often unpredictable ways. Modifying one gene could have ripple effects throughout the entire genome, leading to unforeseen health problems or developmental abnormalities. We might be solving one problem only to create a host of new ones. This is where the concept of off-target effects comes into play. Current gene editing technologies, like CRISPR-Cas9, aren't perfect. They can sometimes cut DNA at unintended locations in the genome, leading to mutations in unexpected places. While researchers are constantly working to improve the precision of these tools, the risk of off-target effects remains a significant concern, especially when dealing with the germline. The stakes are simply too high to tolerate even a small margin of error when the health and well-being of future generations are on the line. Beyond the technical challenges, there are profound ethical considerations. Who gets to decide which traits are desirable and which are not? What constitutes a genetic defect? These are deeply subjective questions, and there's a real danger of creating a slippery slope towards eugenics, the selective breeding of humans to improve the genetic composition of the population. Historically, eugenics has been used to justify discrimination and oppression against marginalized groups. The idea that some people are genetically superior to others is not only scientifically unfounded, but also morally repugnant. Germline editing could inadvertently revive these dangerous ideologies. leading to a society where individuals are judged and treated differently based on their genetic makeup. Furthermore, the accessibility of germline editing raises serious concerns about equity and social justice. If these technologies become available, they are likely to be expensive and inaccessible to many. This could exacerbate existing inequalities, creating a genetic divide between the wealthy and the poor. The privileged few could afford to enhance their offspring's traits, while the majority would be left behind, further entrenching social and economic disparities. Another concern revolves around the potential for unintended evolutionary consequences. By altering the human germline, we are effectively interfering with the natural processes of evolution. While this may seem desirable in the short term, it's difficult to predict the long-term effects on our species. Genetic diversity is essential for adaptation and survival. By artificially narrowing the gene pool, we could make ourselves more vulnerable to new diseases or environmental challenges. Imagine a future where a genetically engineered population is highly susceptible to a novel virus. The lack of genetic diversity could allow the virus to spread rapidly and devastate the population, while a more genetically diverse population might have individuals with natural resistance, allowing them to survive and rebuild. These are not hypothetical scenarios. They are real possibilities that must be carefully considered before we embark on the path of germline editing. The decision to alter the human germline is not one that should be taken lightly. It's a decision that will have profound and lasting consequences for our species and for the future of life on Earth. Therefore, a cautious and ethically grounded approach is paramount. Robust regulations, transparent public discussions, and ongoing scientific research are essential to ensure that germline editing is used responsibly and for the benefit of all humanity, not just a select few. The secrets held within the human genome demand our utmost respect and consideration, especially when contemplating changes that echo through the corridors of time. The future of our species may very well depend on the choices we make today. All right, let's talk about something truly revolutionary: genome editing. For decades, scientists dreamed of precisely altering the genetic code, snipping out flaws and pasting in corrections. That dream is now a reality, thanks to a suite of powerful tools, the most famous of which is undoubtedly CRISPR-Cas9. Think of the human genome as an incredibly complex instruction manual, and these genome editing tools as highly specialized word processors. They allow us to target specific sequences within that manual, make precise changes, and hopefully rewrite faulty instructions. CRISPR-Cas9, which stands for Clustered, Regularly Interspaced short palindromic repeats and CRISPR-associated protein. 9 is borrowed from bacteria. In bacteria, CRISPR-9 is a defense mechanism against viruses. When a virus attacks, the bacteria incorporates snippets of the viral DNA into its own genome, creating a genetic memory of the infection. If the same virus attacks again, the bacteria uses this memory to recognize and destroy the virus. Scientists realized that this system could be adapted for genome editing in other organisms, including humans. Here's how it works. We design a short RNA sequence called guide RNA that matches the DNA sequence we want to target. This guide RNA is then attached to the Cas9 enzyme, which acts like a pair of molecular scissors. The guide RNA leads the Cas9 enzyme to the target DNA sequence. Once there, Cas9 cuts both strands of the DNA. Now, the cell's own DNA repair mechanisms kick in. There are two main pathways for repair. The first, called non-homologous end joining, or NHEJ, is a quick and dirty repair process. It simply glues the broken ends back together, but often introduces small insertions or deletions of DNA bases in the process. This can disrupt the gene, effectively knocking it out or turning it off. This is useful if we want to disable a gene that is causing a problem. The second repair pathway, called homology-directed repair, or HDR, is more precise. If we provide the cell with a DNA template that has the desired sequence, along with the CRISPR-Cas9 system, the cell will use this template to repair the break. This allows us to introduce specific changes into the genome, correcting mutations, or adding new genes. CRISPR-Cas9 isn't perfect, though. One of the biggest challenges is off-target effects. This is when the guide RNA binds to a sequence in the genome that is similar but not identical to the intended target. This can lead to unintended cuts and edits at other locations in the genome, which could have unpredictable and potentially harmful consequences. Researchers are constantly working to improve the specificity of CRISPR-Cas9 and minimize off-target effects by developing new versions of the Cas9 enzyme or using more sophisticated guide RNA design strategies. While CRISPR-Cas9 gets most of the attention, it's not the only genome editing tool available. Other methods, such as TALENs or transcription activator like effector nucleases and zinc finger nucleases or ZFNs, also allow for precise DNA editing. TALENs and ZFNs are proteins that are engineered to bind to specific DNA sequences and cut the DNA at the target site, relying on the cell's own repair mechanisms to fix the break. TALENs and ZFNs FNs, are generally considered to be more specific than earlier versions of CRISPR-Cas9, meaning they are less likely to cause off-target effects. However, they are also more complex to design and produce, which has made CRISPR-Cas9 the more popular choice in many applications. Each of these technologies has its own strengths and weaknesses, and the choice of which tool to use depends on the specific application. So, what are the potential applications of genome editing? The possibilities are vast and span a wide range of fields, from medicine to agriculture to basic research in medicine. Genome editing holds immense promise for treating genetic diseases. Imagine being able to correct the mutations that cause cystic fibrosis, sickle cell anemia, or Huntington's disease. Clinical trials are already underway to test the safety and efficacy of CRISPR-based therapies for these and other genetic disorders. Beyond treating genetic diseases, genome editing could also be used to fight infectious diseases such as HIV and cancer. Researchers are exploring ways to use CRISPR to disable viruses or to engineer immune cells to target and destroy cancer cells. In agriculture, genome editing can be used to improve crop yields, enhance nutritional value, and make plants more resistant to pests and diseases. For example, scientists have used CRISPR to develop rice varieties that are more resistant to bacterial blight, and tomatoes that have a longer shelf life. In basic research, genome editing is a powerful tool for studying gene function. By selectively knocking out or modifying genes, researchers can gain a better understanding of their role in biological processes. This can lead to new insights into human health and disease. However, with great power comes great responsibility. The potential of genome editing raises a number of ethical concerns. One of the most controversial issues is the use of genome editing to modify the human germline, that is, the DNA in sperm, eggs, or embryos. Changes made to the germline would be passed down to future generations, raising concerns about unintended consequences and the potential for eugenics. Many scientists and ethicists believe that germline editing should be approached with extreme caution, if at all. There is a general consensus that it should only be considered for serious genetic diseases where there are no other treatment options, and only after careful consideration of the risks and benefits. Another ethical concern is the potential for genome editing to exacerbate existing social inequalities. If genome editing technologies become widely available, there is a risk that they will only be accessible to the wealthy, leading to a genetic divide between the rich and the poor. It's important to ensure that these technologies are developed and used in a responsible and equitable manner, with careful consideration of the ethical, social, and legal implications. Public discourse and engagement are crucial to inform policy decisions and shape the future of genome editing. As we move forward, it's essential to balance the promise of these technologies with the need to protect human health and well-being, and to ensure that they benefit all of humanity. The human genome, that intricate instruction manual for building and operating a human being, holds far more than just the secrets to our individual traits and predispositions. Embedded within its 3 billion base pairs lies a rich history, a detailed chronicle of our species' evolutionary journey across millennia. By analyzing the subtle variations in our DNA, scientists are piecing together the puzzle of human origins, tracing our ancestral migrations and uncovering the genetic adaptations that allowed us to thrive in diverse environments. This section delves into the fascinating realm of evolutionary genomics, exploring how the human genome serves as a powerful time capsule, revealing the story of our shared past. One of the most compelling insights from genomic analysis is the confirmation of our shared ancestry with other primates, gorillas, and other related species. We can identify the genetic changes that occurred along the evolutionary lineage leading to modern humans. These comparative studies reveal the genes that underwent significant alterations, potentially contributing to uniquely human traits such as bipedalism, increased brain size, and complex language. For instance, researchers have pinpointed specific genes involved in brain development and function that show accelerated evolution in humans compared to other primates. These genes may have played a crucial role in the emergence of our cognitive abilities. Furthermore, the genome provides evidence for our African origins. Genetic diversity is highest among African populations, reflecting the longer period of human habitation and evolution on the African continent. As humans migrated out of Africa, they carried only a subset of this original genetic diversity, leading to decreased variation in populations found in other parts of the world. By studying the patterns of genetic variation across different populations, scientists can reconstruct the migratory routes our ancestors took as they spread across the globe. These genomic breadcrumbs paint a vivid picture of human dispersal, confirming the out-of-Africa theory of human origins. But the story doesn't end there. As human populations settled in new environments, they faced unique challenges that required genetic adaptation. Genomic analysis has revealed numerous examples of local adaptation, where specific gene variants became more common in certain populations due to their beneficial effects in a particular environment. For example, the ability to digest lactose, the sugar found in milk, is much more prevalent in populations with a long history of dairy farming. This adaptation allowed these populations to exploit a valuable food source, providing them with a nutritional advantage. Similarly, genes involved in skin pigmentation show strong evidence of adaptation to different levels of ultraviolet radiation. Populations living in regions with high sun exposure evolves darker skin to protect against UV damage, while populations in regions with low sun exposure evolves lighter skin to allow for greater vitamin D. Synthesis, another fascinating area of research, involves the study of ancient DNA. By extracting DNA from ancient human remains, such as bones and teeth, scientists can directly analyze the genomes of our ancestors who lived thousands of years ago. This ancient DNA provides a unique window into the past, allowing us to track genetic changes over time and to understand how human populations have mixed and interacted throughout history. One of the most remarkable findings from ancient DNA research is the discovery of interbreeding between modern humans and other hominin species, such as Neanderthals and Denisovans. Most modern humans of non-African ancestry carry a small percentage of Neanderthal DNA in their genomes, a testament to the encounters and interbreeding that occurred between our species tens of thousands of years ago. This interbreeding may have even provided modern humans with advantageous genes that help them adapt to new environments. Genomic analysis also sheds light on the genetic basis of human diseases and health disparities. By comparing the genomes of individuals with and without specific diseases, scientists can identify genes that increase the risk of developing those conditions. Understanding the genetic factors that contribute to disease can lead to new diagnostic tools, targeted therapies, and personalized prevention strategies. Moreover, genomic studies can help explain why certain diseases are more prevalent in some populations than others. These disparities may be due to differences in genetic background, environmental factors, or a combination of both. By unraveling the complex interplay between genes and environment, we can work towards reducing health disparities and improving health outcomes for all. The insights gained from evolutionary genomics are not just of academic interest. They have profound implications for our understanding of human nature, our place in the natural world, and our future as a species. By tracing our ancestry and uncovering the genetic adaptations that shaped our history, we can gain a deeper appreciation for the diversity and resilience of humankind. Disparities can pave the way for more effective and equitable healthcare. However, it is crucial to approach evolutionary genomics with caution and sensitivity. Genetic information can be misused to promote harmful stereotypes or to justify discriminatory practices. It is essential to remember that genetic variation is continuous and that there is no clear dividing line between different human populations. We are all members of the same species, sharing a common ancestry and a common destiny. The goal of evolutionary genomics should be to promote understanding, empathy, and respect for human diversity, not to reinforce harmful divisions. In conclusion, the human genome is a treasure trove of information about our evolutionary history. By analyzing the subtle variations in our DNA, scientists are unraveling the story of human origins, tracing our ancestral migrations, and uncovering the genetic adaptations that allowed us to thrive in diverse environments. These insights have profound implications for our understanding of human nature, our place in the natural world, and our future as a species. As we continue to explore the secrets of the human genome, we can expect even more fascinating discoveries that will further illuminate the complex and remarkable journey of humankind. All right, let's dive into the fascinating world of comparative genomics. It's a field that essentially boils down to this: understanding ourselves by comparing our genome to the genomes of other species. Sounds simple, right? But the insights we gain are anything but. At its heart, comparative genomics is about identifying similarities and differences in the genetic makeup of various organisms. By aligning and analyzing these genomes, we can piece together evolutionary relationships, pinpoint genes that are crucial for specific functions, and even understand the genetic basis of diseases. Think of it like this. Imagine trying to understand how a car engine works without ever seeing another engine. You could figure some things out, sure, but wouldn't it be easier if you could compare it to a truck engine, a motorcycle engine, and maybe even a lawnmower engine? You'd start to see patterns, common components, and unique adaptations that reveal how each engine is designed to work. That's exactly what comparative genomics does for the human genome. We compare it to everything from single-celled organisms to complex mammals, and the results are often surprising. One of the earliest and most impactful findings was just how similar we are to other creatures, at least on a genetic level. For instance, humans share around 98% of our DNA with chimpanzees. That's a pretty staggering number when you consider the obvious differences between us. It highlights that even small genetic changes can have significant effects on an organism's development and characteristics. But it's not just about finding similarities. The differences, those seemingly small variations, are where much of the evolutionary magic lies. By identifying regions of the genome that are highly conserved across species, meaning they've remained relatively unchanged over millions of years, we can infer that these regions are likely to be functionally important. These conserved sequences often encode essential proteins or regulate critical developmental processes. If a sequence is so vital that evolution hasn't tinkered with it much, chances are it's doing something pretty important. Conversely, regions of the genome that show rapid evolution or are unique to certain species can provide clues about adaptations that allow those species to thrive in specific environments. Take, for example, the genes involved in lactose tolerance in humans. Most mammals lose the ability to digest lactose after infancy. However, in some human populations, particularly those with a long history of dairy farming, mutations arose that allowed adults to continue producing lactase, the enzyme that breaks down lactase. By comparing the genomes of lactose tolerant and lactose intolerant individuals, as well as the genomes of other mammals, scientists could pinpoint the specific genetic changes responsible for this adaptation. This not only tells us about human evolution, but also provides insights into how our genes interact with our diet and environment. Comparative genomics also sheds light on the origin and spread of diseases. By studying the genomes of pathogens, like viruses and bacteria, we can trace their evolutionary history, identify the genes responsible for virulence, and even develop new strategies for combating them. For example, comparing the genomes of different strains of influenza virus allows scientists to track the emergence of new variants and predict which strains are most likely to cause epidemics. This information is crucial for developing effective vaccines and antiviral drugs. Similarly, comparing the genomes of antibiotic-resistant and antibiotic-sensitive bacteria helps us understand the mechanisms of antibiotic resistance and develop strategies to slow its spread beyond medical applications. Comparative genomics helps us understand the fundamental principles of biology. By comparing the genomes of organisms with different body plans, we can identify the genes that control development and determine how these genes have been modified over evolutionary time to produce the incredible diversity of life on Earth. For instance, the Hox genes are a group of genes that play a crucial role in determining the body plan of animals, from insects to humans. These genes are arranged in a specific order on the chromosome, and their order corresponds to the order of body segments they control. By comparing the Hox genes of different animals, scientists have gained insights into how body plans have evolved and diversified. Now, you might be wondering, how is all this actually done? Well, the process typically involves several steps. First, the genomes of the organisms of interest are sequenced. Then, these sequences are aligned to identify regions of similarity and difference. This alignment process can be computationally intensive, especially when dealing with large and complex genomes. Once the genomes are aligned, researchers can analyze the data to identify conserved regions, rapidly evolving regions, and genes that are unique to certain species. They can also use statistical methods to infer evolutionary relationships and identify genes that are associated with specific traits or diseases. Powerful computers and sophisticated software are essential tools in this field. The availability of complete genome sequences for a wide range of organisms has revolutionized comparative genomics. The more genomes we have to compare, the more comprehensive and accurate our understanding of evolution and biology becomes. Databases like GenBank and the Ensemble Genome Browser provide access to a vast amount of genomic data, making it easier than ever for researchers to conduct comparative analyses. However, comparative genomics also faces some challenges. One major challenge is the complexity of genomes themselves. Genomes are not just linear strings of DNA. They are complex, three-dimensional structures with intricate regulatory networks. Understanding how these structures and networks function requires more than just comparing DNA sequences. It requires integrating information from other sources, such as gene expression data, protein interaction data, and epigenetic data. Another challenge is the difficulty of interpreting the functional significance of all the genetic variation we observe. Just because a gene is different between two species doesn't necessarily mean that this difference is responsible for any observable phenotypic difference. It could be a neutral mutation that has accumulated over time. Despite these challenges, comparative genomics remains one of the most powerful tools we have for understanding the human genome and our place in the tree of life. It provides a framework for interpreting the vast amount of genomic data that is being generated and for translating this information into practical applications in medicine, agriculture, and conservation. As technology continues to advance and we sequence more genomes, comparative genomics will undoubtedly play an even greater role in shaping our understanding of the world around us and the intricate workings of life itself. Okay, let's talk about the microscopic universe teeming within us, the microbiome. It's easy to think of ourselves as purely human, defined solely by the roughly 20,000 genes nestled within our cells. But the reality is far more complex and fascinating. We are, in essence, walking ecosystems, hosts to trillions of microorganisms, bacteria, fungi, viruses, and archaea, collectively known as the microbiome. And these tiny tenants aren't just freeloaders. They play crucial roles in our health, our development, and even our behavior. Now, where does the genome come into play? It's all about interaction. Think of it as a constant conversation between our human genes and the microbial genes residing within us. We provide them with a home, nutrients, and a stable environment. In return, they provide us with a vast array of benefits, many of which are directly influenced by the genes they carry. Consider this. The human genome encodes for a limited number of enzymes capable of breaking down complex carbohydrates. But our gut microbiome, it's a carbohydrate digesting powerhouse. The genes within these microbes encode for thousands of enzymes that can break down dietary fiber, plant cell walls, and other complex sugars that we, as humans, simply can't process on our own. This allows us to extract energy and nutrients from food that would otherwise pass right through us. These microbial genes, in essence, extend our own metabolic capabilities. But it's more than just digestion. The microbiome is deeply involved in the synthesis of essential vitamins. Think about vitamin K, vital for blood clotting, and several B vitamins. Many of these are synthesized primarily by bacteria residing in our gut. We rely on these microbial factories, directed by their own unique genetic instructions, to produce these vital nutrients that our own genes don't code for. So, in a very real sense, the genes of our microbiome are supplementing our own genetic deficiencies. The connection goes even deeper, influencing our immune system. From birth, our immune system learns to distinguish between self and non-self. But it also needs to learn to tolerate the beneficial bacteria within our gut. This delicate balance is crucial for preventing autoimmune diseases and chronic inflammation. The interaction between our immune system genes and the genes expressed by our microbiome helps to train and calibrate our immune response. Certain microbial genes produce molecules that directly interact with immune cells, modulating their activity and promoting tolerance. Disruptions to this delicate balance, often caused by factors like antibiotics or dietary changes, can lead to immune dysregulation and a host of health problems. The gut-brain axis is another fascinating example. The microbiome communicates with the brain through a complex network involving the vagus nerve, the immune system, and the production of neurotransmitters. Microbial genes influence the production of neurotransmitters like serotonin, dopamine, and GABA, which play critical roles in mood, behavior, and cognitive function. Alterations in the gut microbiome have been linked to anxiety, depression, and even neurodegenerative diseases. While the exact mechanisms are still being unraveled, it's clear that the genes of our microbiome can profoundly impact our brain health. So, how do we study this complex interaction between our genes and those of our microbiome? It involves sequencing all the DNA present in a sample, such as a stool sample, to identify the different types of microorganisms present and the genes they carry. By analyzing the metagenome, we can get a snapshot of the functional potential of the microbiome and how it might be influencing our health. Another approach is to study the metabolites produced by the microbiome. Metabolites are small molecules that are produced as a result of microbial metabolism. By analyzing the metabolites present in blood or urine, we can get insights into the activities of the microbiome and how it is interacting with our host. This field, known as metabolomics, complements genomics and provides a more complete picture of the microbiome-host interaction. The Human Microbiome Project was a landmark initiative that aimed to characterize the microbial communities living in and on the human body. This project generated a vast amount of data that has been invaluable for understanding the role of the microbiome in health and disease. It revealed the sheer diversity of the microbiome and highlighted the significant variation between individuals. But there are challenges. The microbiome is incredibly complex, and it varies significantly from person to person. Factors like diet, lifestyle, genetics, and environmental exposures all influence the composition and function of the microbiome. Untangling the complex interactions between these factors is a major challenge. Another challenge is that many of the microorganisms in the microbiome are difficult or impossible to culture in the lab. This makes it difficult to study their function and to develop targeted therapies. However, advancements in sequencing technologies and computational methods are helping us to overcome these challenges. The potential applications of microbiome research are vast. We are already seeing the development of probiotics and prebiotics designed to promote a healthy gut microbiome. Fecal microbiota transplantation, FMT, which involves transferring the stool from a healthy donor to a recipient, has shown promising results in treating recurrent Clostridium difficile infection and is being investigated for other conditions as well. Looking ahead, personalized microbiome therapies are likely to become more common. These therapies would be tailored to the individual based on their unique microbiome profile and genetic background. Imagine a future where we can use genetic information to predict an individual's risk for certain diseases, and then use targeted microbiome interventions to prevent or treat those diseases. That the interaction between our human genes and the genes of our microbiome is a complex and fascinating area of research. It's a reminder that we are not simply individuals, but complex ecosystems. By understanding the intricate interplay between our genes and the genes of our microbial partners, we can unlock new insights into human health and develop innovative strategies for preventing and treating disease. The secrets within the microbiome are only beginning to be revealed, and they hold the key to understanding ourselves in a whole new light. Welcome back, everyone. In this segment, We're shifting our focus from the genes themselves to the dynamic interplay between our genetic code and the world around us. We're diving into environmental influences and how these external factors interact with our genes to shape our health and disease outcomes. It's crucial to understand that our genes don't operate in a vacuum. They're not a static blueprint dictating a predetermined fate. Instead, they're more like a musical score, a set of instructions that can be interpreted in countless ways depending on the conductor. In this case, the environment. Think of it this way. You might inherit a gene that predisposes you to a certain condition, say type 2 diabetes. But whether that gene actually manifests as the disease depends largely on factors like your diet, exercise habits, and exposure to environmental toxins. The gene loads the gun, but the environment pulls the trigger. So, what exactly constitutes the environment in this context? It's an incredibly broad term, encompassing a whole range of factors. We're talking about everything from the air we breathe and the food we eat, to the social and economic conditions we live in, and even our exposure to infectious agents. One of the most well-studied areas of environmental influence is nutrition. What we eat has a profound impact on our gene expression. The process by which our genes are turned on or off, and how much protein they produce. For example, a diet high in processed foods and saturated fats can trigger inflammatory pathways, activating genes that promote inflammation and increasing the risk of chronic diseases like heart disease and certain cancers. Conversely, a diet rich in fruits, vegetables, and whole grains can activate genes that promote detoxification, reduce inflammation, and protect against cellular damage. Beyond diet, our physical activity levels also play a significant role. Exercise isn't just about burning calories and building muscle. It also has a powerful effect on our genes. Regular physical activity can activate genes involved in glucose metabolism, improving insulin sensitivity and reducing the risk of type 2 diabetes. It can also stimulate the production of brain-derived neurotrophic factor, or BDNF, a protein that promotes brain health and protects against cognitive decline. Exposure to environmental toxins is another critical factor. We're constantly bombarded with a cocktail of chemicals in our air, water, and food, many of which can disrupt our hormonal systems, damage our DNA, and increase the risk of various diseases. For example, exposure to air pollution has been linked to increased rates of respiratory illnesses, heart disease, and cancer. Similarly, exposure to pesticides and other chemicals in our food supply has been associated with developmental problems, reproductive disorders, and certain types of cancer. But it's not just about the external environment. Our internal environment, including our gut microbiome, also plays a crucial role in shaping our gene expression. The trillions of bacteria, fungi, and other microorganisms that live in our gut produce a vast array of metabolites that can interact with our genes, influencing everything from our immune function to our brain health. A diet rich in fiber and fermented foods can promote a healthy gut microbiome, while a diet high in processed foods and antibiotics can disrupt the balance of our gut flora, leading to inflammation and increased susceptibility to disease. The fascinating field of epigenetics is where environmental influences exert their effects on our genes. Epigenetics literally means above or on top of genetics, and it refers to the mechanisms that control gene expression without altering the underlying DNA sequence. Think of it as a series of switches that can turn genes on or off, or dim them up or down in response to environmental cues. One of the most common epigenetic mechanisms is DNA methylation, where chemical tags are added to DNA, typically silencing gene expression. Another is histone modification, where proteins called histones, around which DNA is wrapped, are modified, altering the accessibility of genes and affecting their expression. These epigenetic changes can be influenced by a wide range of environmental factors, including diet, stress, and exposure to toxins, and perhaps even more remarkably, some of these epigenetic changes can be passed down from one generation to the next, meaning that our experiences and exposures can potentially impact the health of our children and grandchildren. This is the concept of transgenerational epigenetic inheritance, and it's a rapidly evolving area of research with profound implications for our understanding of health and disease. Consider the Dutch hunger winter, a period of severe famine in the Netherlands during World War II. Studies have shown that individuals who were exposed to famine in utero had a higher risk of obesity, heart disease, and other health problems later in life. These effects were linked to epigenetic changes that altered gene expression in ways that promoted fat storage and increased insulin resistance. Similarly, studies have shown that early childhood adversity, such as abuse or neglect, can lead to epigenetic changes that increase the risk of mental health problems, substance abuse, and chronic diseases later in life. These epigenetic changes can affect the development of the brain and the regulation of the stress response system, making individuals more vulnerable to the negative effects of stress. Understanding the interplay between genes and the environment is crucial for developing effective strategies for disease prevention and health promotion. It highlights the importance of adopting healthy lifestyles, reducing exposure to environmental toxins, and addressing social and economic disparities that can impact health outcomes. It also underscores the need for personalized medicine approaches that take into account both genetic and environmental factors when tailoring treatments and interventions. While we can't change the genes we inherit, we can control many of the environmental factors that influence their expression. By making informed choices about our diet, lifestyle, and exposure to toxins, we can take proactive steps to optimize our health and reduce our risk of disease. This is the power of understanding the environment's role in shaping our genetic destiny. It's not about blaming our genes for our health problems, but about empowering ourselves to take control of our well-being by creating an environment that supports optimal gene expression. And that's a message with a potential to transform public health. The future of genomics is not just about faster sequencing or cheaper tests. It's a radical reimagining of how we understand and interact with life itself. We're standing on the cusp of a new era, fueled by emerging technologies that promise to reshape medicine, agriculture, and even our understanding of human evolution. One of the most exciting frontiers is long-read sequencing. Traditional sequencing methods chop DNA into short fragments, making it difficult to assemble complex or repetitive regions of the genome. Long-read sequencing, on the other hand, can analyze DNA segments tens of thousands of base pairs long in a single read. This allows scientists to map entire genes, identify structural variations with greater accuracy, and resolve previously inaccessible parts of the genome. Imagine being able to see the whole picture instead of just scattered pieces of a puzzle. Then, there's single-cell genomics. Our bodies are made up of trillions of cells, each with its own unique genetic profile. Single-cell genomics allows us to analyze the DNA, RNA, and proteins within individual cells, providing unprecedented insights into cellular diversity and function. This is particularly crucial for understanding diseases like cancer, where subtle genetic differences between cells can drive tumor growth and drug resistance. By analyzing individual cancer cells, we can identify the specific mutations that are fueling their growth and develop targeted therapies that are more effective and less toxic. Another revolutionary technology is spatial genomics. While single-cell genomics tells us what's happening inside individual cells, spatial genomics reveals where those cells are located within a tissue or organ. This is vital for understanding how cells interact with each other and with their environment. Imagine being able to see a map of a tumor showing not only the genetic makeup of each cancer cell, but also its position relative to blood vessels, immune cells, and other components of the tumor microenvironment. This level of detail could revolutionize cancer diagnosis and treatment, allowing us to target specific cells in specific locations. Artificial intelligence and machine learning are also playing an increasingly important role in genomics. The human genome is vast and complex, containing billions of data points. AI algorithms can sift through this data, identify patterns, and make predictions that would be impossible for humans to do alone. These algorithms can be used to identify genes that are associated with disease risk, predict how patients will respond to different treatments, and even design new drugs. The ability to quickly process and interpret massive data sets is accelerating genomic research at an unprecedented rate. CRISPR-Cas9, the gene editing technology that has captured the world's attention, continues to evolve. While its initial applications focused on correcting genetic defects, researchers are now exploring its potential for a wide range of other applications, including developing new diagnostic tools, creating disease models, and even enhancing human traits. The precision and versatility of CRISPR-Cas9 are transforming the landscape of genetic engineering, opening up possibilities that were once considered science fiction beyond these specific technologies. The future of genomics also hinges on data, sharing, and collaboration. As more and more people have their genomes sequenced, the amount of genetic data available is growing exponentially. To fully realize the potential of this data, it needs to be shared openly and securely among researchers around the world. This requires developing new data privacy protocols, establishing standardized data formats, and fostering a culture of collaboration within the scientific community. Only by working together can we unlock the secrets of the genome and translate them into tangible benefits for society. But the future of genomics is not without its challenges. As we gain a deeper understanding of our genetic makeup, we also face complex ethical and societal questions. How do we protect genetic privacy in an age of ubiquitous data collection? How do we ensure that genomic technologies are used equitably and do not exacerbate existing health disparities? How do we address the potential for genetic discrimination in employment, insurance, or other areas of life? These are questions that require careful consideration and open dialogue. Moreover, the increasing accessibility of genetic information raises questions about personal autonomy and informed consent. Do people fully understand the implications of having their genomes sequenced? Are they prepared for the potential emotional and psychological impact of learning about their genetic predispositions to disease? Are they able to make informed decisions about their healthcare based on this information? Education and counseling are essential to ensure that people can make responsible choices about their genetic information. The societal impact of genomics extends far beyond the realm of medicine. In agriculture, genomic technologies are being used to develop crops that are more resistant to pests, diseases, and climate change. This could help to improve food security and reduce the environmental impact of farming. In forensics, DNA sequencing is used to identify criminals, exonerate the innocent, and trace human ancestry. In conservation biology, genomics is being used to study endangered species, understand their evolutionary histories, and develop strategies for protecting them. As genomics becomes more integrated into our lives, it is important to remember that genes are not destiny. While our genes can influence our risk of developing certain diseases, they do not determine our fate. Our environment, lifestyle, and personal choices also play a critical role in shaping our health and well-being. Genomics should be used to empower individuals to make informed decisions about their health, not to stigmatize or discriminate against them. Ultimately, the future of genomics is about empowering individuals and communities to live healthier, longer, and more fulfilling lives. It's about harnessing the power of genetic information to prevent disease, personalize treatment, and improve the quality of life for all. But it's also about ensuring that these technologies are used responsibly, ethically, and equitably. The genome is a shared inheritance, and its secrets should be used for the benefit of all humankind. As we move forward, we must proceed with caution, humility, and a deep respect for the complexity and interconnectedness of life. The potential is enormous, and the responsibility is even greater, even with all the incredible progress we've made in genomics. It's vital to remember that we're still just scratching the surface. We've deciphered the code, but truly understanding the language of the genome presents a host of formidable challenges. These aren't just technical hurdles. They also touch upon the ethical and societal implications of wielding such powerful knowledge. One of the biggest limitations is interpreting the sheer complexity of genomic data. We now have the ability to sequence entire genomes relatively quickly and cheaply. But turning that sequence, that string of As, Ts, Cs, and Gs into meaningful insights about health, disease, and even behavior is an immense task. Imagine receiving a massive instruction manual in a language you barely understand. You might be able to identify some words and phrases, but grasping the overall message and how each part contributes to the whole would be incredibly difficult. That's essentially where we are with the human genome. We can see it, but we don't fully understand it. This challenge is compounded by the fact that genes don't operate in isolation. They interact with each other in complex networks, and these interactions can be influenced by environmental factors, lifestyle choices, and even chance. It's like trying to understand a symphony by only listening to one instrument at a time. You might get a sense of the melody, but you'd miss the richness and complexity of the full orchestra. Furthermore, much of the genome is still shrouded in mystery. We often hear about the roughly 2% of our DNA that directly codes for proteins. But what about the other 98%? For a long time, it was dismissed as junk DNA. However, we're now realizing that much of this non-coding DNA plays crucial roles in regulating gene expression, essentially turning genes on and off, or fine-tuning their activity. Understanding these regulatory mechanisms is essential for understanding how the genome functions as a whole and how variations in these regions can contribute to disease. Figuring out the functions of this dark matter of the genome is a major area of ongoing research. Another significant hurdle is the lack of diversity in genomic research. The vast majority of genomic studies have been conducted on individuals of European descent. This means that our understanding of genetic variation and its impact on health is heavily skewed. What might be a common genetic variant in one population could be rare or even absent in another. This lack of diversity can lead to inaccurate risk assessments and ineffective treatments for individuals from underrepresented populations, exacerbating existing health disparities. Addressing this requires a concerted effort to include diverse populations in genomic research and to develop analytical methods that are sensitive to the unique genetic backgrounds of different groups. Beyond the technical challenges, there are also significant ethical considerations. Genetic information is incredibly personal and sensitive. It can reveal information not just about an individual, but also about their family members. This raises concerns about privacy, discrimination, and the potential for misuse of genetic data. For example, could employers or insurance companies use genetic information to discriminate against individuals who are predisposed to certain diseases? Could genetic testing be used to select for or against certain traits in embryos, leading to unintended social consequences? These are complex questions that require careful consideration and robust ethical frameworks. The promise of personalized medicine tailored to an individual's genetic profile is incredibly exciting. But it also presents practical challenges. Translating genomic findings into clinical practice requires sophisticated diagnostic tools, targeted therapies, and healthcare professionals who are trained to interpret and apply genetic information. It also requires a fundamental shift in the way we approach healthcare, moving from a one-size-fits-all model to a more individualized and preventative approach. Gene therapy. The potential to correct genetic defects by introducing new or modified genes into cells holds immense promise for treating inherited diseases. However, it is still a relatively new field with significant risks. Ensuring the safety and efficacy of gene therapy treatments is paramount. We need to develop better delivery methods, minimize the risk of off-target effects, and carefully monitor patients for long-term complications. Finally, The use of genome editing technologies like CRISPR-Cas9 raises profound ethical questions about the potential to modify the human germline, that is, to make changes to the DNA that are passed down to future generations. While germline editing could potentially eliminate inherited diseases, it also raises concerns about unintended consequences, the potential for misuse, and the long-term impact on the human gene pool. There is broad consensus that germline editing should only be considered in cases where there is a clear medical need and where there are no reasonable alternatives. Furthermore, any such interventions should be subject to rigorous ethical oversight and public debate. In conclusion, while genomics has revolutionized our understanding of life, significant hurdles remain in fully harnessing its potential. Overcoming these technical, ethical, and social challenges is crucial to ensuring that genomic knowledge is used responsibly and equitably to improve human health and well-being for all. The journey to fully decode and apply the secrets of the human genome is a marathon, not a sprint, and requires a collaborative effort from scientists, ethicists, policymakers, and the public alike. The Human Genome, that incredible instruction manual for life, holds not only the secrets to who we are, but also the keys to preventing disease. While we often hear about genes causing illnesses, the reality is far more nuanced. Our genetic code provides invaluable information that, when properly understood and applied, can empower us to proactively safeguard our health and dramatically reduce our risk of developing certain conditions. Think of it this way: your genome is like a weather forecast. It can't predict the future with 100% certainty, but it can identify potential storms on the horizon. Armed with this knowledge, you can take steps to reinforce your defenses, seek shelter, and mitigate the potential damage. In the context of disease, this means using genetic information to identify predispositions, adopt preventative measures, and personalize healthcare strategies. One of the most direct applications of genomic information in disease prevention lies in identifying individuals at high risk for specific inherited conditions. For example, genes like BRCA1 and BRCA2 are strongly linked to an increased risk of breast and ovarian cancer. Genetic testing can reveal whether an individual carries mutations in these genes, allowing them to make informed decisions about preventative measures. These might include more frequent screenings, prophylactic surgeries like mastectomies or oophorectomies, or lifestyle modifications to minimize other risk factors. Similarly, genetic screening can identify individuals at increased risk for cardiovascular diseases like familial hypercholesterolemia, a genetic disorder that leads to high cholesterol levels. Early identification allows for aggressive management of cholesterol levels through diet, exercise, and medication, significantly reducing the risk of heart attacks and strokes. For other inherited conditions, like cystic fibrosis or sickle cell anemia, genetic testing can identify carriers, allowing them to make informed reproductive choices and potentially prevent the transmission of these diseases to future generations. This proactive approach, informed by genetic insights, shifts the focus from reactive treatment to preventative care. A paradigm shift with profound implications for public health. But the story doesn't end with inherited conditions. Our genes also influence our susceptibility to a wide range of complex diseases, like type 2 diabetes, Alzheimer's disease, and certain types of cancer. While these diseases are not solely determined by genetics, our genes can significantly increase or decrease our risk. Understanding these genetic predispositions can empower individuals to take proactive steps to mitigate their risk. For instance, if genetic testing reveals an increased risk for type 2 diabetes, an individual can adopt lifestyle modifications, like maintaining a healthy weight, exercising regularly, and following a balanced diet. These seemingly simple interventions can often be remarkably effective in preventing the onset of the disease, even in individuals with a strong genetic predisposition. Similarly, research is uncovering genetic variants that influence the risk of Alzheimer's disease. While there is currently no cure for Alzheimer's, understanding an individual's genetic risk can encourage them to adopt lifestyle changes that promote brain health, such as engaging in mentally stimulating activities, maintaining a healthy diet, and managing cardiovascular risk factors. While these measures may not completely eliminate the risk of developing Alzheimer's, they may delay the onset of the disease or slow its progression, improving the quality of life for affected individuals. Furthermore, genomics is revolutionizing the field of cancer prevention. We're moving beyond simply identifying higher risk genes like BRCA1 and BRCA2, and are beginning to understand how multiple genes interact with each other and with environmental factors to influence cancer risk. This knowledge is paving the way for personalized cancer prevention strategies tailored to an individual's unique genetic profile. This might involve targeted screening programs, chemo prevention with specific medications, or lifestyle interventions designed to minimize exposure to known carcinogens. Pharmacogenomics, the study of how genes affect a person's response to drugs, is also playing an increasingly important role in disease prevention. By understanding an individual's genetic profile, doctors can choose the most effective medications and dosages, minimizing the risk of adverse side effects and maximizing the therapeutic benefits. This is particularly important for medications used in preventative therapies, such as statins for cholesterol management or antiplatelet drugs for preventing blood clots. Moreover, advances in genomic sequencing are enabling us to identify novel biomarkers for early disease detection. Biomarkers are measurable indicators of a biological state or condition. By analyzing an individual's genome, we can identify genetic variants that are associated with early stages of disease, even before symptoms appear. This allows for earlier diagnosis and intervention, improving the chances of successful treatment and preventing the progression of the disease. Of course, the application of genomic information in disease prevention is not without its challenges. One of the biggest hurdles is the complexity of gene-environment interactions. Many diseases are influenced by a complex interplay of genetic and environmental factors, making it difficult to predict an individual's risk with certainty. Furthermore, genetic testing can raise ethical concerns about privacy, discrimination, and psychological distress. It's crucial to ensure that genetic information is used responsibly and ethically, with appropriate safeguards in place to protect individual rights. Despite these challenges, the potential of genomics to revolutionize disease prevention is undeniable. As our understanding of the human genome continues to grow, we will develop even more sophisticated tools for identifying at-risk individuals, personalizing preventative strategies, and ultimately creating a healthier future for all. The era of preventative genomics is upon us, and its potential to transform healthcare is truly remarkable. By harnessing the power of genetic information, we can move beyond treating diseases to preventing them in the first place, creating a future where individuals are empowered to take control of their health and well-being. So, we've explored the incredible progress made in understanding the human genome. We've mapped its vastness, deciphered portions of its code, and even begun to manipulate its very structure. But it's crucial to acknowledge that the journey is far from over. The human genome remains, in many ways, an enduring mystery, peppered with unsolved questions and tantalizing hints of future discoveries. Think of it like this: we've assembled a complex machine, the human body, and we've obtained the instruction manual, the genome. But the manual is incomplete, written in a language we're still learning, and contains sections we don't yet fully understand. We know the basic functions of many parts, the genes, but the precise roles and interactions of countless other components, the non-coding regions, remain shrouded in shadow. One of the biggest mysteries revolves around the non-coding DNA, often referred to as junk DNA, a term that's increasingly recognized as a misnomer. While it doesn't directly encode proteins, which are the workhorses of our cells, it constitutes the vast majority of the genome. We're slowly uncovering its crucial role in regulating gene expression, influencing when, where, and to what extent genes are activated. But the precise mechanisms and the full extent of its influence are still largely unknown. How do these regions, once dismissed as useless, orchestrate the intricate symphony of gene activity that determines our development, physiology, and susceptibility to disease? That's a question that continues to drive research. Another area of active investigation is the interplay between our genes and the environment. We know that our genes don't operate in a vacuum. External factors such as diet, lifestyle, and exposure to toxins can significantly impact gene expression, a field known as epigenetics. These epigenetic modifications, chemical tags that attach to our DNA, can switch genes on or off, influencing our health and well-being. But understanding the complex dance between our genes and the environment, and how these epigenetic changes are inherited across generations, remains a significant challenge. How can we leverage this knowledge to promote healthier lifestyles and prevent disease? Furthermore, while genome-wide association studies have identified numerous genetic variants linked to various diseases, pinpointing the exact causal genes and understanding the underlying mechanisms are often incredibly difficult. Correlation does not equal causation, and identifying the true drivers of disease amidst the noise of genetic variation is a complex undertaking. Many diseases are influenced by multiple genes, each contributing a small effect, further complicating the picture. We are learning more about gene interactions, or how genes act together to influence phenotypes. However, this is one of the most challenging tasks facing genomic researchers. And consider the microbiome that reside within our bodies. These microbes possess their own genomes, which interact with our own in complex and often poorly understood ways. The composition of our microbiome can influence our digestion, immune system, and even our mental health. But disentangling the intricate interactions between our genes and the genes of our microbial inhabitants, and understanding how these interactions contribute to health and disease, is a burgeoning field with immense potential. How can we harness the power of the microbiome to improve our health and treat disease? Looking ahead, the future of genome research is brimming with promise. Emerging technologies such as long-read sequencing, single-cell genomics, and advanced imaging techniques are providing unprecedented insights into the structure, function, and regulation of the genome. These tools are allowing us to examine the genome at a higher resolution than ever before, revealing previously hidden details and complexities. Artificial intelligence and machine learning are also playing an increasingly important role in genome research. These powerful algorithms can analyze vast datasets of genomic information, identify patterns, and predict gene function with remarkable accuracy. They can also help us to develop personalized treatments tailored to an individual's unique genetic profile. Gene editing technologies, such as CRISPR-Cas9, hold immense potential for correcting genetic defects and treating inherited diseases. But these technologies also raise profound ethical considerations, which we discussed earlier, regarding the safety and potential consequences of altering the human germline. As we move forward, it is crucial to engage in open and informed discussions about the responsible use of these powerful tools. Finally, the integration of genomic information with other forms of data, such as electronic health records, imaging data, and environmental exposure data, is paving the way for a more holistic understanding of human health. This systems biology approach recognizes that our genes are just one piece of a much larger puzzle, and that understanding the interplay between our genes, our environment, and our lifestyle is essential for preventing disease and promoting well-being. The journey to fully understand the human genome is a marathon, not a sprint. There will undoubtedly be setbacks and challenges along the way, but with continued innovation, collaboration, and a commitment to ethical principles, we can unlock the secrets of the genome and harness its power to improve human health and well-being for generations to come. The story of the genome is far from over. In fact, it has only just begun. The greatest discoveries are yet to be made.